Hey, welcome back. Today we are going to talk about cofactors and inhibitors. So many of the enzymes that we are working with or will work with require non-protein helpers called cofactors. So these cofactors generally fit right into the active site. You see that here. And they attract the substrate or give the substrate a better fit. Either way, they are necessary, for the enzyme to function properly. So again, here's our enzyme. Its shape is everything. So a lot of times we need that cofactor to just sit right in the active site, and that attracts the substrate or allows the substrate to fit in there nice and tight. A lot of times cofactors are metals like zinc or iron or copper. Cofactors can be organic, which means they're carbon-based. That's like the macromolecules. They can be like lipids or like nucleotides or like sugars. When this is the case, they're called specifically coenzymes. So cofactors are necessary for an enzyme to attract the substrate. When that cofactor is carbon-based, it's called a coenzyme. Vitamins. You've always heard, hey, take your vitamins. Well, vitamins oftentimes function as coenzymes. They help your enzyme to better attract or better fit the substrate they work on. Very, very, very common, very popular cofactor is hemoglobin. Well, actually, hemoglobin is a protein that uses a cofactor, so better stated that way. Very important protein is hemoglobin. Technically, it's not an enzyme. I know this whole unit's on enzymes. Technically, hemoglobin doesn't speed up any reactions, so it's not technically an enzyme. But it's still a protein that still uses a cofactor, and that's what this lesson's about, so we'll take a little closer look. So hemoglobin, the protein, and here it is. It's made up of 4 subunits. See those here, the 2 alpha chains and the 2 beta chains. Hemoglobin is a protein found in red blood cells. And its whole job, its whole responsibility, is to carry oxygen. And that's how red blood cells carry oxygen around to all the parts of your body. It does this by having the cofactor iron. So within all hemoglobin atoms, all hemoglobin proteins, you have iron. And that's what actually holds on to, grabs, attracts, and holds on to oxygen. It's indeed the iron that gives your blood its red color. So that's why blood is red, is because of all the iron found in blood. Again, the iron is the cofactor, hemoglobin is the protein, the iron is what attracts the oxygen, which is what hemoglobin is responsible for carrying. That brings us to inhibitors. So inhibitors are almost like the opposite of cofactors. Inhibitors block enzymes from working with their substrate. So you see that in the picture here. Enzymes can be competitive or non-competitive. A competitive inhibitor competes for the same spot. It's going to lock on exactly in the active site. So here's the normal enzyme. It's going to bind with the substrate, and that's normal. Here is a picture of competitive inhibition. So here's our competitive inhibitor, and again, you can see it's competitive because it's competing with the substrate for the actual active site. It's actually taking up the active site, and it's blocking or inhibiting the substrate from coming in there, and now the enzyme can't work. And that's where they get competitive inhibition from. They're named competitor because they're competing for the active site. Non-competitive inhibitors, so you see that over here on the right, they don't bind at the active site. They bind at a different spot on the enzyme. So you can see this one. This non-competitive inhibitor has bonded in the back of the enzyme, and what that does is it changes the shape of the whole enzyme. It distorts the active site, and now the substrate can no longer bind, so we can't have that reaction take place, And that's all due to a non-competitive inhibitor. And they're called non-competitive inhibitors because they don't directly compete. They're non-competitive. They're not trying to get to that spot first. They're just coming in the back and changing the whole shape and changing the whole game. Non-competitive inhibitors. Penicillin. We talked about this drug already, several times actually. Penicillin is an inhibitor. So first of all, on the left here, we see normal bacteria cell, this gray protein. Its job, so here are pieces of the cell wall. So the green and teal colors, those are glycogen. So those are the plant parts, or the plant-like parts of the wall, the carbohydrates, and then these little circles are amino acids. And what happens in a normal bacteria cell is this gray protein is going to link the amino acids of one part of the wall to the next. And now they're tied together, and your wall is strong, or the bacteria cell's wall is strong, and it's linked, and that's what that protein, that gray protein, is in charge of doing. On the right, we see the same process starting. There's that gray protein, it's starting to link together pieces of the wall, but the drug penicillin, which are these orange peas, represented by the orange peas over here, is an inhibitor. The penicillin is going to bind with that protein, which prevents it from binding with the amino acids and building that bridge between the wall. So when this is bound by penicillin, it's inhibited, this enzyme is inhibited from doing its job, and that's why bacteria cell walls fall apart. They aren't built correctly, they aren't linked together in the presence of penicillin. It's acting as an inhibitor to block the production or the formation of bacteria cell walls, and that's how the drug penicillin works. It's an inhibitor. Alright, so that's our lesson on cofactors and inhibitors. Take a look at the homework. Have a good weekend.